So good afternoon, and thank you to the audience, first of all, for attending the webinar today. Today we have the webinar of the Developmental Biology and Genomic Modification Group. This group is coordinated by Berta Alsina. She's working at the Universitat Pompeu Fabra. So thank you, Berta, for organizing the webinar. And if you want, you can introduce the speaker today.
Okay, so thank you very much, Mar, for coordinating this. Can you hear me?
Yeah.
Yes. Okay. So today we have the pleasure to have Anna Bigas. She's the senior or the coordinator of the program of cancer research at IMIM. She did her PhD in 1993, and then she moved to the States for her postdoc in the Fred Hutchinson Cancer Center in Seattle, and there she did a very important contribution working on notch signaling and hematopoiesis. So she has been focusing in this aspect for many years. Then she came back. She became a junior group leader at IRO, and and then she moved as senior group leader at IMIM. And recently, she also has been the scientific director of CIPF, and also she's group leader and deputy director of the Institut Josep Carreras, because she has some work more on basic hematopoiesis, but also her work also is very related to leukemia and cancer. So Anna, she's a very well recognized researcher in this field. She contributes with being panel of the ERC, the ANEP, and many other organizations, international organizations. She has published in very good journals because she has done very good contributions on the role of stem cells in hematopoiesis, notch, and Wnt signaling. And I hope that today we will be able to follow a little bit some of this work. And also she is reviewer of some of these top journals. And she's also EMBO member from 2014, plus many other awards and grants and so on. So I'm very pleased that you have accepted this invitation, Anna, and I'm sure we will enjoy this webinar. Thank you very much.
Thank you, Berta. It is actually a pleasure for me to be a speaker today here for the society. So I'm going to just share my screen. Hopefully you will see it now. If everything is okay, I will start. So what I want to talk about today is our work from the last years on understanding how the hematopoietic stem cells are made. And this is something that happens, this is an event that happens during embryonic development, as many of the cell types in our body, they get specified during this time when we are embryos. So this is the hierarchical tree of hematopoiesis. It could be from many other tissues, mainly having the stem cells at the top of the hierarchy. And we know a lot about these stem cells in the adult life. We know that they live in at least for mammalian systems, they live in the bone marrow. They are well protected there inside the bones. But what we're going to talk today is where are the first ones of these hematopoietic stem cells made, and what do we know about how they specify during this time in development. Basically, this is especially referring to the mouse embryonic development. We get very early hematopoietic cells that come from extraembryonic tissues around day 7, day 8 of development, and mainly coming from the yolk sac, we get the first sites, or the first signs of hematopoietic cells that mainly form erythrocytes and macrophages. However, at this time, when we first see these cells, we cannot detect any cell that we can classify as hematopoietic stem cells. So basically, the hematopoietic development goes, I like to say, goes in waves, and these waves are kind of backward. So first, there is differentiated cells, differentiated functional cells produced, probably to serve the embryo when grows enough and cannot transport oxygen by diffusion, and it needs some specialized transport and other needs. And then in the second wave, the progenitors of different lineages are specified. And this, we know it happens mainly still in the extraembryonic tissues, still in the yolk sac, but also in the allantoids. And these progenitors are also specified from endothelial cells that actually transform into hematopoietic cells. This is a transition that we call, we know as EHT, for endothelial to hematopoietic transition. However, although at this stage there's many cell types that are made, and we can actually find many of the similar adult counterparts, we cannot find hematopoietic stem cells, as we will see in a second, what we call hematopoietic stem cells. It is later on around, in the mouse around day 10, and in the human is around week 4 of development, when we find, or we can detect, the first sites of hematopoietic stem cells. And we can detect them in this region, which is in the aorta, in the dorsal aorta of the trunk of the embryo, and that is surrounded by gonad and mesonephros tissues. And this is why it's called AGM, for aorta gonad mesonephros. And just zooming into the aorta at this time of development, we find the same type of EHT, the same type of endothelial to hematopoietic transition that we also find in the extraembryonic tissues. So some specialized endothelial cells that we call them hemogenic, they are able to make this transformation of shape and transcriptome and become hematopoietic cells. And some of these cells, we know now that they are hematopoietic stem cells. This is a very well conserved process in development, and actually, at least for the vertebrate species, they mostly have similar cells, similar type of development into endothelial cells transitioning into the hematopoietic. And for example, studies in the zebrafish, in the Hermomel lab, but also in Travers' lab, showed how some of these cells that you can see here by the FLIC1 reporters, so endothelial cells, are rounding up in the dorsal aorta, and in the case of the zebrafish, they travel into the cardinal vein, where they will go to the CHT region that is equivalent to the fetal liver in the mouse. So this was pretty much the first time that we could visualize that actually endothelial cells were transitioning to this hematopoietic state. And obviously with the zebrafish, this was an advantage to visualize all this process. But so what happens in the mouse? What can we see in the mouse? The mouse is a little more complicated to do imaging. We will see later what we've done in the lab to try to image these cells, but it's not so easy because we cannot image the whole embryo, the whole organism. So we mainly most of the work comes from immunos, in situ, or whole mount immunostainings. This is, for example, a transversal section from Roshana, who's a postdoc in the lab. And what we can see here, this is a transversal section of the dorsal aorta at day 10 of development of a GFI1 reporter mouse embryo in this case. And GFI1 is actually one of the first genes expressed in these hemogenic endothelial cells in the dorsal aorta. That's what we see here. We see the endothelium, but then we see this red nuclear staining that is the GFI1 reporter, and is underlying, or it's highlighting the cells that are in the endothelium that will become or will have the potential to become hematopoietic. And this is conserved also in the clusters because these cells form clusters of cells that emerge from this dorsal aorta at this time of development. And what we can see on the right, we see these clusters now marked with another very useful marker, which is c-KIT. And c-KIT is also one of the first surface markers that is expressed in these hematopoietic clusters that appear in the aorta. And these are whole mount immunostainings. And we can see how all these clusters distribute mainly in the ventral side of the dorsal aorta. So basically what we have seen is that in the mouse embryo, if we looked at the right time of development in this embryonic aorta, we see these endothelial cells that have the capacity to form these hematopoietic clusters that we can see here with the c-KIT marker. In the last years, we have known quite a lot of how to classify and how to study these hematopoietic clusters. And we know now that they are not homogeneous, they are very heterogeneous, they are formed by very different types of cells. And we can actually identify them because they conserve some of the endothelial markers, but also acquire all the different types of hematopoietic markers. As we have seen, the GFI1 would be one of the first ones, and then c-KIT would be labeling the whole cluster, but also CD41, and at the later stage, the CD45 classic pan-hematopoietic marker would appear and would characterize the hematopoietic system. And how do we know that these cells are stem cells and not the ones I showed you that appear in the yolk sac or in the other extraembryonic tissues? Well, because early studies by dissecting mouse embryos, dissecting different parts of the embryo, and dissociating cells and transplanting them into irradiated mice, demonstrated that only from this tissue, only from this aorta tissue in the AGN, was the first one and the only one at this time of development that was able to reconstitute the hematopoiesis of an irradiated mouse. So this is how we know that the stem cells, first the stem cells we can detect with the meaning stem cell, meaning a cell that is able to reconstitute the hematopoietic system, are first originated here. So this is what we are studying in the lab. This is some of the questions that we have been trying to answer during this year. So we know that the stem cells are made at this time of development, but we know very little about what are the signals that dictate this hematopoietic fate from the endothelium, and which signals dictate actually that some of these cells in the clusters are stem cells. But as I will show you, not all of them are stem cells, only a few of them are stem cells. And obviously the ultimate goal that we would like we and many others would like to be able is to generate this type of cells in vitro. And so one of the questions that we had some time ago is how were these clusters made? And we know that they appear, as we have seen, in the dorsal part, in the ventral part of the dorsal aorta, and they have, so we can see with many different shapes and many different types of clusters, and we know that they are heterogeneous and they are formed by different types of cells. So when Cristina Porcheri was a postdoc in the lab, what she did, she set up different techniques to actually visualize or be able to visualize these cells as they were coming out of the aorta. And what she did with help of a group in Rotterdam, there's the Zerza group in Rotterdam and Catherine Robin, they actually had showed that having this slicing this embryo in slices that were about 100 nanometers, 100 micrometers, and putting them in agarose to immobilize them, and then keeping them at the right conditions, you could actually be able to have an overnight time-lapse video of these slices. So basically, we were slicing the embryo at the site where we know these clusters appear. We would be looking for the right sections where we could see that there were cells that were seemed to be able to produce the hematopoietic clusters, and then focusing this for the overnight video and hope that the cells would do something. And actually, this was pretty nice. We can see here one reporter. This was, in this case, a CD41 reporter. So most of the cells in the cluster are in yellow, and DAPI and the H2B GFP, sorry, is in blue. So we could see how cells were dividing. For example, we can see several mitoses in this cluster. So this is one of the first observations that these clusters were actively proliferating. And we could also measure some of the parameters, that we will see in the clusters. So that was very useful. Actually, one of the first observations that Cristina could do was to show that the first thing that happened to make these clusters was cell division. We would see these cells, these single cells that were bulging, and then the next thing in any single cluster that she could image at this stage was first division, and most of the time there were a second division. So we would have these cells having one or two divisions. That's the first event that was happening for cluster formation. So this kind of pointed to the fact that these clusters were clonal. So they would be one cell would divide and they would form the different progeny would form the cluster. And this was one of the questions we tried to address in the lab. And what Cristina did was to use the confetti system, the confetti mice, in which in this case, we crossed with the recombinase reporter for under the control of V cadherin. So what we got from these crossings were randomly labeled endothelial cells with the different fluorescent proteins. And in this case and in these conditions, this was a CRE-ERT. Sorry about some of these labels that have been cannot be read. But we could see that within this system, we could titrate down the label of the cells and have individual cells in the endothelium of the aorta that were labeled with different colors. And so now what Christina wanted to know is, okay, from these labeled cells, what are they doing in a specific time, period of time? Are they dividing and getting monoclonal clusters so they will all are same color? Or are they, or what we have there is a polyclonality and we have different colors? So what we saw, and this is the summary of a lot of work that she did, because she had to screen lots of embryos to actually get some of this, get this data. But that was clear that what she saw in all the clusters that were in the 2-3 cell stage were monoclonal. So as we had seen by imaging, these cells divide, and they obviously, if they divide, if they are colored, the progeny of this division is the same color. However, once these clusters were larger, when they grew to 4, 5, or even larger clusters from 6 to 12 cells, we started seeing that there were unlabeled cells, but also clusters with different colors, with cells from different colors, indicating that some other contribution of cells that were not labeled originally, or that were labeled with another color, was happening in the clusters. So what we knew, and with a lot of work, this is just to tell the end of the story. We saw that actually neighboring cells in a cluster, after a cluster reached a certain size, were indeed getting recruited by the cluster. And we also could see that these cells that were getting recruited into the cluster were hemogenic cells. So at the original site of the cluster divides, but then the hemogenic cells surrounding the cluster are being recruited into the cluster. And this indeed was very interesting to see, to understand the composition of the clusters and to try to understand how are these hematopoietic stem cells being specified. So I'll come back to this later, because we have done a lot of work trying to understand what are the signals that actually regulate these clusters and the size, and whether these cells are permitted to go into this hematopoietic cluster or not. But one of the main questions, in fact, is with all these clusters, we know that not all the cells are stem cells. Not all are going to be hematopoietic stem cells. And in fact, if we look at the literature and the earlier studies, mainly done in the Mèdiques and Gerzerak's lab, what we see is that very few of these clusters, or very few of these hematopoietic cells in the cluster, are able to reconstitute the hematopoiesis of unirradiated mice. And in fact, when they have done calculations, for example, here we can see at day 11 of development, this is a very detailed study and actually very laborious, trying to quantify the hematopoietic stem cells detected in an embryo. We could see that at day 11, this number is as low as 1. And this is still true. I mean, this study is from 2002, but this is still true. If we, like Mèdiques likes to say, if we divide an AGM into 3 mice and transplant, only 1 will get reconstituted. And so we know that many of these cells that are produced in the clusters are not able to be stem cells, or they are not stem cells yet, because we also know that if we actually put them in explants or give them some signals, many of these cells, then we can amplify this number of stem cells that we can reconstitute the mice. So we think that at this time of development, there's very few that actually have this capacity of hematopoietic stem cells, but probably there's many more that can make it. And also clonal fate mapping studies, both in zebrafish and in the mouse, have estimated by different color labeling of hematopoietic stem cells. They have estimated that there are more than one, or many more than one stem cell in the embryo, which probably makes sense. So many of these cells will in the end make it, although at this time of development we cannot detect it. So from zebrafish in the Zones lab, what they came up was with that there were from 20 to 30 hematopoietic stem cells being formed per fish, per embryo. And in the mouse, the McKinney-Freeman lab came out with around around 500 hematopoietic stem cells that are currently being made. But in any case, this is much fewer than the cells that we find in the clusters. So one of the things that we have tried to understand is what are the signals that define the composition of the clusters, and what are the signals that affect the amount of hematopoietic stem cells that can be found in a cluster. And this is from a review that we wrote with Elaine Zerzak a couple of years ago. And obviously the pathways involved in this process are similar as the classical pathways that we can probably find in any single tissue. And they are important or mainly important during the embryonic development. But in the lab, we have worked, as Berta said at the beginning, we have worked with this pathway for many years, and we have tried to understand what are what was the role of Notch in this specific process, and how was this controlling hematopoietic stem cell development. So we know that there's this is a section of the embryonic aorta, and this is a staining for the active Notch1 protein, and we can see that not all the cells in the aorta are positive for active Notch1, but we have a variety of cells with different levels of Notch and different stainings for Notch that are actually clearly positive. So one of the stories that we have worked in the recent years was trying to understand exactly this cluster formation, what was controlling the amount of cells in the cluster and the recruitment of cells in this cluster. And we actually took advantage of blocking antibodies against the Notch ligands, because this is not a new reagent, but I would say that now it's a kind of a very useful reagent that we have been able to implement into these studies. So what we can do with these antibodies, because mainly because a lot of these proteins, a lot of the notch pathway genes and proteins are involved in many decisions, and when we have to work with genetic manipulation, there's always genetic manipulation at a specific time of development, and there is not really a specific CRE for this early hematopoietic event. So we normally have to use endothelial CREs. And then some of these elements are also important in endothelial specification, as we will see. So the antibodies, the blocking antibodies have been great. We can use them. We can inject them in the beating heart of the embryo when we take the embryo, and then we can incubate. We can have this couple 2-3 hour incubation within the embryo, and then analyze what happens in a very short period of time. So in this case, we saw that the blocking delta-4 was crucial to understand what was happening with the clusters, because we didn't, after 5 hours of blocking with the delta-4 incubation, we didn't perturb the cell cycle. So cell cycle was exactly the same in these short period incubations. But what we did see was that suddenly, instead of having a small cluster cells distributed around the aorta, we were having a huge amount of cells that were piling up in these hematopoietic clusters. And here we are looking at the c-KIT marker, as I showed you before. This is in the clusters. So this is the quantification of many embryos of c-KIT positive cells per cluster. And you can see that in general, there are more cells, but then there are a few of these clusters actually are very, very huge when we block delta-4. And what we saw was that the number of positive cells, KIT positive cells per cluster was increasing. Sorry. But there were not the number of clusters. So the number of clusters remained the same, but these clusters actually had more cells and were growing without getting more proliferation. And this is, in fact, what it brought us to test and to understand what was happening with this recruitment of cells that were next to the clusters. In fact, what Roshana in the lab, what she did using this GFI1 reporter and doing this same strategy of incubating for a few hours with the blocking delta-4 antibody, she could show how she got more of these homogenic GFI1 KIT positive cells now within the clusters, while these homogenic KIT negative cells, so the cells that have not been in the cluster yet, are decreasing. So we actually imaged these cells moving from the endothelium into the cluster, and this was happening all when delta-4 was blocked. So this was clearly pointing to that, or demonstrating that Notch was involved in controlling the cells, where they were, and how they went into the clusters. And the final question is, well, is this what's happening when the cells go into the clusters? Is this useful? Can we? What is the effect of this on the hematopoietic stem cells? And this was addressed just by getting these cells after blocking with this antibody and transplanting into irradiated mice. And what we could see that early cells that have very low repopulation capacity, when we incubated them with the blocking delta-4 antibody, they had increased capacity to reconstitute the hematopoietic system of these irradiated mice. So this is the summary of this, and basically what we showed was that these hemogenic cells, they moved into clusters or into already initiated clusters, and this decision is controlled by delta-4 and notch, and we had many more data to show that this was a notch-dependent decision. And by blocking delta-4, what we are doing is just enhancing this recruitment of cells into the clusters massively. And probably what this is making is just maturing the stem cells, or maturing potential early cells into the ones that have this hematopoietic capacity. So we're just gonna go a little bit back because, okay, as I showed you, notch can control the cells that go, or when the cells go into the clusters, and by these ways controlling the maturation or the time of maturation of these cells into the stem cells. However, we know much more of what notch is doing. Notch is not only doing that in this specific system. We know that there is, especially in vertebrates, we have several notch receptors, several notch ligands, and there is a very specific or very well balanced timing with all the signals of this pathway. Mainly, as I'm sure all of you know, the notch receptor sits at the cell surface and interacts with different ligands that can be either from the delta family or from the jagged family, and they actually have specific different requirements. When these interactions occur, the proteases with the gamma secretase complex cleave the several protease cleavages, and then the intracellular domain of notch is able to move to the nucleus, participate, interact with coactivators, and participate in active transcription. And these in general will create, by different means and by different models that have been postulated in different model organisms, will have will make a whole homogeneous populations of cells to have different variety of cell fates, as it can be seen by lateral inhibition or by lateral induction. But mainly the result will be that we'll have from an homogeneous population will make up an heterogeneous population of cells. And as I said, we have different receptors, we have different ligands in vertebrates, and we know that from other early studies from our lab, but also from many other labs, that several of these are involved in the hematopoietic stem cell fate acquisition. We actually know that not only delta-4, as I showed you, that was modeling the cluster formation and the amount of cells that were getting into the clusters, but also jagged is important. Sorry, jagged is important to actually get active notch signal that will make transcriptional targets that will be expressed in these hematopoietic cells, and that some of them are crucial to get this hematopoietic fate, such as GATA2. We also know that many of these notch elements are shared by the endothelial cells to make other endothelial decisions, such as the arterial development. And because we know that delta 4, we just saw how it's important to make the clusters, but we knew before how delta 4 is crucial to make arterial development. But also Notch1 is the same receptor, and this is shared between the arterial cells, endothelial arterial cells, and the hematopoietic cells. So in this way, I think I'm just gonna kind of summarize what we know now about how this is used, how the notch, in this case, Notch1, is shared by both tissues, the endothelial and the hematopoietic. And depending on the availability of the ligand, it actually can go either more into the hematopoietic or favor the hematopoietic phase or favor the arterial phase. And we know that this is just how the interaction between, or how the structure between the interaction of ligand and receptors can have a very different type of outcome depending whether it's interacting with one or another. And we're just trying to figure out or to understand in different systems what this outcome can be. We know that if we delete jagged, there are no HSCs, no hematopoietic stem cells. This is early work. We know that if we delete jagged, we also have a lot of higher notch activity in the endothelium. So in a way, jagged is also titrating the amount of notch activity. We know that if we block delta 4, we have more HSCs, and we have in these cells we have lower notch activity. And this is basically what I just told you, but with some of the results that we have collected over the years. And we also know that we require notch to get the transcriptional activation of both Hess transcription factors, which are the main targets of notch, but at the same time, this hematopoietic specific transcription factor, which is GATA2 and is required for hematopoietic stem cell emergence. And here, this feed-forward loop, what it does is regulate very tightly the levels of GATA2, and it all depends on the first signal, which is Notch1. I think I'm just gonna summarize, go to the end, and basically I wanted also to make this comparison with the endothelium sprouting and the cluster formation, because as you can see, these patterns are repeated in development, probably in many of the types of branching or cells going into different directions. And in fact, for the endothelial sprouting, this Raf Adams and Rui Benedito's group, they showed some years ago how actually there was this delta 4 positive cells, this delta 4 that was signaling from the tip cell to the stalk cell to actually activate Notch1 and prevent this cell to go to move forward into the tree of the angiogenic tree. And we think that similar things are happening with the clusters, how delta 4 positive cells are signaling the cells that are underneath in the endothelium to maintain high levels of notch and prevent these cells to go up to the cluster. However, when jagged comes, probably this equilibrium is broken, and then other interactions are happening there. I'm not. This is the summary of all these different interactions that I showed you, how this is controlling the amount of cells in the clusters and how hematopoietic stem cells are, or how many hematopoietic stem cells, or at which point the hematopoietic stem cells are being developed. And just some couple of slides just to tell you that what we are trying to do is to implement some of these into the embryonic stem cells and how to make these cells. We know for many years that these cells can differentiate in vitro, and they can make hematopoietic cells of many cell types in vitro. But there has not been a robust way to make hematopoietic stem cells from these embryonic stem cells. These cells, for somehow they differentiate, they make lots of progenitors, but we cannot keep them, or we cannot make them as undifferentiated hematopoietic stem cells that can reconstitute the hematopoiesis of irradiated organisms. So this is the big challenge. We are doing screens, and this is some of the screens we're doing in the lab to try to get different types of genes and play with these cells, also putting notch and trying to make these cells engraftable into irradiated mice. And more recently, this is becoming, so we are going from the embryo body of the ES cells into the gastruloid wall in collaboration with Alfonso Martinez Arias here at the UPF. And basically from these ES cells, now we can keep them in a way, or make them to self-organize and kind of form definite patterns during embryonic development, and hopefully get them making some of these endothelial, homogenic endothelium-like cells that hopefully in the future we can try to get some of these hematopoietic stem cells. And just to finish, this is just the acknowledgements to many people, many collaborators that have been crucial in many times of this work. The lab members now that are doing the work, mainly Roshana Tambiraja, but also more recently Susanne van den Brink, and obviously all the other people who have been in the lab who are now in different places of science, and Jordi, Guiu, and Robert Moreno. They were crucial in the early stages of this work, and Cristina Porcheri and Leonor Gama-Norton in other times. Thank you very much, and I'll hope to take your questions.
Okay, so thank you very much, Anna. It has been great. I'm very excited. Yeah. I have not knew some of your last work, so very interesting. I don't know if we have some questions from the chat. I don't see right now. So maybe I can start so people then have time. I have a lot of questions.
Okay, good.
So yes, so these clusters, what I see is that they appear kind of separate one to the other one on the dorsal aorta. So this is kind of a stochastic, you think, or there is also notch involved in the separation and the distance of these clusters?
Yeah, at some point we tried to look for some patterns, whether we could see. We couldn't really see a pattern. In fact, they appear kind of stochastic, but not completely, because they are also very concentrated in the regions where the vitelline artery is joining the dorsal aorta. So there are special location, or there is a special, who knows, physical location or turbulence or physical cues that mechanical cues that may be also influencing that they are mainly formed there. So it's not, they are not everywhere. And this is, I'm sure this is not stochastic, but then when you get into the region, we don't know really. it seems to be areas of GFI1 positive cells, so it seems to be areas of hemogenic endothelium. And maybe are these areas or these competent regions where there is some kind of signal to specify that, but we don't know.
Okay, good. Let me see. Yes,
I have some curiosities, Berta. If you I just want to remember that the people can do the questions in the chat. But I'm not from the field, but you have been saying, Anna, that you are all interested in culturing these hematopoietic stem cells. And I would like if you can comment which are the applications that you are thinking for these hematopoietic stem cells.
Well, this has been the holy grail for many years, because if you can make, so first stem cell therapy is the bone marrow transplant. This has been on for more than 50 years now. It's cured many leukemic patients or patients with other genetic problems in the blood system. So if you can get a replacement of your hematopoietic system, if you have a problem in your hematopoietic system and you can get a good replacement, this is one of the easiest treatment. But what happens? That not many people have compatible donors. So especially depending on which ethnic groups, there's very few donors. There is also other applications that go into gene therapy, replacement of gene editing in the future, who knows, for specific diseases. And if you can get hematopoietic stem cells, these would probably solve lots of problems than using primary cells. If there was a possibility to get hematopoietic stem cells from embryonic stem cells, or IPS, this would simplify things, protocols. It would be much easier, because it's easier to culture these cells than to have primary cells, have them in culture, and get all these possible problems, differentiation problems. But so there's several applications with now with the immunotherapy and all these other products that we can get from blood. So this is something that people are directly obtaining differentiated cells. But if you have hematopoietic stem cells, you can that's a much safer source for starting the differentiation than getting embryonic stem cells, for example. So there's several applications. Thanks for the question.
Commenting about that. So I imagine that I think that most of the stem cells, they tend to have kind of a small toolkit that identifies them as stem cells. So in many different types of stem cells, we tend to have the same transcription factors and molecules, like SOX, OCT, and then probably GFI1 or PO. So have you done some transcriptome, like pointing out maybe some few transcription factors that you don't need to go to organoids and maybe just reprogram the embryonic stem cells? People done that, or it's...
There's a couple of reports out there with reprogramming from fibroblast, going from fibroblast into hematopoietic stem cells. There's endothelial cells going to hematopoietic stem cells, and also from ES cells with the specific transcription factors. Each one. So I recall these three examples. They have all been very very good papers, very recognized work, but very hard to reproduce in the different labs for some reason. We don't know how to make these cells can be made. They have been made in the past, but there's something missing. There's something that makes them that you can get them at a specific point, but then it's hard to.
They don't survive, maybe.
So there's all these cocktails, because and also we have tried, and in fact part of our screening is trying to get cocktails of cells that then will go into repopulate cells in the mouse, and yeah, this has happened. I mean, in many different labs, they publish this cocktail, they get nice results specifically at some point, but then you try to make the same genes in your cells and they don't work, or you don't get them. So it's still something missing.
And I have another question. So these stem cells that you find in the embryo, so are the same during the whole life? Do they kind of change? The stem cells from one stage to adult are different? Do they age? They live forever? Things like this?
Yeah. They do all kinds of things. So I didn't go further than the AGM, right? We looked at the very specific period of time. And actually, I didn't go into the fact that after the AGM, these cells go to the fetal liver, at least for mammalian organisms, and in the caudal region in the fish, no? But the fetal liver in mouse. And there we know there is a lot of things happening. I mean, I think they get mature, but then they get different signals there that probably make them. So we are in the lab, we are now working a lot on dormancy. We know that some of them get more dormant or more capable of being dormant. And this starts as early, or we can see that there's signals that do that already in the fetal liver. We know there are dormant hematopoietic stem cells in the adult. In fact, in the mouse, there are cells that are you don't even require these cells for the life of the mouse. You only require them when you serially transplant these mice. But sometimes some of these stem cells, we know they are dormant for pretty much the life of the mouse. We don't know in the human because we have not been able to look at this so closely, but in the mouse is clear. And so there are different types of stem cells in the adult, and we think at least, and we have some data on that some of these characteristics are acquired probably in the fetal liver or at these different stages of development.
Mm-hmm.
Yeah.
The correlation with aging is very interesting, Berta. This that you have pointed out is really interesting, and probably you can find differences, no, Anna? If you see.
Yeah, we haven't looked into that, but so we know there are stem cells that can live longer than others, or that are more preserved than others, and probably this is crucial in an aging situation. Mm-hmm. And we think some of these potency, let's put it this way, it's acquired during development, or some specific instructions seem to affect the capacity of these cells. So that's probably also very important in an aging situation. We know that with aging, we have more stem cells, or more phenotypically stem cells, but they are less potent. They have a bias to go into myeloid lineage, doing less lymphoid cells, more myeloid cells. And this happens normally in aging. So the fact that these cells can do, these stem cells can get this different capacity for differentiation or for recovery, I think it's very important. It's probably even more important in the human than it is in the mouse. So.
Very interesting.
Yeah, I have just a final question. So you talked about Delta 4. You put the antibody against Delta 4, but you didn't explain why you were looking Delta 4. You had previous idea because I know that you have been studying many notch ligands and things like this. So it just was kind of
Delta 4 came into the picture because, in fact, the Delta 4 knockouts have a very, very strong endothelial phenotype. So nobody has been able to look at the hematopoiesis because they don't make hematopoiesis. And then if you delete Delta 4 in the endothelium, it's the same problem that the total knockout had before. It's you cannot time it so well. So basically at the end you have nothing. When we look for expression, we've done a lot of expressions and immunostainings in different times of the lab history. And when there were starting to be good Delta 4 antibodies, we actually look at the expression of Delta 4, and we knew it was, or it had been described, it's in the arterias. We all use it as an arterial marker. Yes, but we saw this huge Delta 4 expression in the clusters, and we couldn't understand what was going on. So when the antibodies came, I think that was one of we wanted to test what was happening with Delta 4 in the clusters, and yeah, there was this interesting phenotype that suddenly we got bigger clusters.
Yeah,
so
very beautiful.
We kind of follow that to understand how were the clusters formed. But
yeah, and the balance between Delta 4 and JAK is also interesting because they are competing from the endothelial cells, no? So I guess.
Everything. When you look, this is we've done a lot of expression. Now we also have single cell data with the transcriptome, and also we've done, I didn't present, we've done some index sorting using, now there's these antibodies, some of these antibodies work quite well by flow cytometry. So there's more tools, there's many more tools now that now we can use to characterize these cells. But still from the immunostaining, you have you see the reconstruction of where these cells are, and everything, so we have jagged, we have delta, we have notch 1, we have notch 4. Later on there is notch 2. So there is a lot of different elements, and this is part of why it's quite difficult to know what this is doing, because it's kind of hard to separate the functions of each one, no? So we still can do some more notch work. Probably.
Okay, I see that there is no questions. I think everybody's very quiet here, but I guess they have been following us.
But in any case, we are more or less we have been more or less 1 hour, so I think that it's time to close. And in any case, if any of the people in the audience wants to do any questions, just remember that Anna is open to
sure.
You.
Just email me.
Yeah. Okay, so thank you very much, Berta and Anna, for this interesting webinar. And thank you to the audience also for attending the webinar. And I hope to see all of them again at the end of the month, that we have another one with Marisol. join us. So thank you, Berta. Thank you, Anna.
Thank you, Anna, very much.
Thank you, Berta. Thank you, Mar. That was great.